Hi students, welcome you all to another important session of Brainiacs Capsule. Okay, so today we are going to discuss the chapter Biomolecules from Chemistry. It's a very important chapter in the sense that all organic chemistry that we are learning are applied in the chapter Biomolecules. Okay, so we are going to discuss three important questions from this chapter. The first one is with respect to the topic proteins. Okay, so I hope you could see the screen. The question here is, which of the following are considered as essential amino acids? Okay, we know that amino acids are the important units of proteins. We can say proteins are made up of amino acids. Mainly we can say alpha amino acids. Amino acids can be alpha, beta, gamma, delta amino acids. Okay, but in our body proteins are mainly made up of alpha amino acids. Okay, so this alpha amino acids, if you see its fundamental structure, it looks like what actually? So I can write here an alpha amino acid will look like this. Okay, so this is an alpha amino acid. Okay, I hope you could see. Okay, so if you look at the fundamental unit of an alpha amino acid, it will have a carbon. Okay, then there will be one hydrogen unit will be present here. Okay, and there will be one I can say there is a COOH unit would be there. Okay, and there is something called an amino group would be present. Okay, and next here we can have an alkyl group or even hydrogen atom. So here different amino acids, they will differ in this R group actually. Okay, and we can say one important thing. These amino acids, if you see here, they are CO with the presence of COOH group, we can say they are acidic in nature. Okay, because of the presence of NH2 group, we can say they are also basic in nature. So we can say based on the R group, sometimes the R group has more NH2 groups, sometimes it has COOH group. Based on that, we can say the amino acid is acidic or basic. And if it has equal number of COOH and NH2 group, we can say they are neutral in nature. And one important property of amino acid is they can exist as Zwitter ion. What is Zwitter ion? We can say that because of the presence of the lone pair that is present in the nitrogen atom, and we have something that is called H plus, which can be liberated by this carboxylic acid. What will happen? It will liberate the H plus over here. Okay, it will liberate the H plus over here, and what will happen? We can see this H plus can attack whom? The lone pair present here. And because of that, what will happen, sir? I can explain you. So once the H plus goes from here, a negative charge would be developed. A negative charge would be developed here, and the H plus will attack the lone pair here. So because of that, this will change to what? I can say this will change to NH3 with a positive charge. Okay, now if you see the amino acid, it has both charges, positive and negative charge. Such a form of existence is called Zwitter ion. Very important, Zwitter ion. So this is some theory part that is required, and we can say these amino acids are classified into two mainly. They are called essential amino acids. I can say, listen, essential amino acids, and the second one we can say non-essential amino acids. What are they? Non-essential amino acids. Now, what is the meaning of essential amino acids? It means this cannot be synthesized inside the human body, and you need to take it in the form of diet. Non-essential means it will be synthesized inside the human body, so you don't need to take it as an extra as part of your diet. Okay, now in this question they are asking which are the famous essential amino acids. So please, it is very important to remember valine, leucine, isoleucine, lysine, then phenylalanine, methionine, threonine, then tryptophan and histidine. These are the various famous essential amino acids. So I used to group it like this. These four I will remember in one stretch: valine, leucine, isoleucine and lysine. Then I will remember it like pre-medical test, PMT. Okay, that is phenylalanine, methionine and threonine. Then you have TH, that is tryptophan and histidine. Okay, so now if you look at the options, you can see here which of the following are essential amino acids. We know tryptophan, lysine, it's an essential amino acid. Histidine, it is an essential amino acid. So I can directly say option 1 is my correct answer. And if you look at the other options, you can see serine. Serine is a non-essential amino acid. So from that itself, you can eliminate option 2, 3 and 4. Clear? And another important point to remember here is almost all amino acids are optically active. That means here the R groups are totally different. So that means all the 4 valencies are occupied by 4 different groups. So I can say these will be normally optically active. Okay, but if you take the amino acid glycine, here the instead of R group you can see a hydrogen is present, and because of that there are 2 similar groups coming on the carbon atom, and because of that I can say it is optically inactive. So one of the optically inactive amino acid, if they are asking, the answer would be glycine, which is the simplest amino acid. Okay, because for R there is only hydrogen is present. I hope you understood. Now let's go to the next question. It's very important. So this is from the topic carbohydrate. So we know that here carbohydrates, we have a lot to learn. I will give you a rough idea what we need to learn in carbohydrates. Listen. So we are supposed to learn the topic carbohydrates. So we are supposed to learn how carbohydrates are classified. So we know that the carbohydrates are classified into monosaccharides. Okay, then you are supposed to learn disaccharides. Okay, disaccharides. Okay, then you are supposed to learn something called polysaccharides. Okay, so monosaccharides, we need to learn the important things over here, mainly glucose. How the scientists, they have discovered glucose and what are the various tests they have performed to discover the structure of glucose. Then we need to learn about fructose. Okay, then if you coming to disaccharides, mainly we are supposed to learn sucrose, very important. So considering sucrose, it is one of the non-reducing sugar. Okay, I will tell you in other section why it is called non-reducing. Other than that, we are supposed to learn lactose and maltose. Okay, and coming to polysaccharides, we are expected to learn here starch. Now what we are going to see is the session on polysaccharides. Actually, starch, we are supposed to learn cellulose and glycogen. So these are the main things you are expected to learn. Okay, so this is just a rough idea. Okay, now in this question they are talking about amylose and amylopectin, and I can say that starch is made up of 2 main components. One is amylose and the next one is amylopectin. Okay, so they are asking what is the difference between the structure of amylose and amylopectin. So this you can answer if you know what fundamentally amylose and amylopectin is. So I can easily say amylose is a hot water soluble component and it constitutes 50 to 20% of starch. So amylose is contributing 50 to 20%, and amylose is a long unbranched chain. There is no branching in amylose, which is made up of 200 to 1000 units of which one? Alpha D glucose units, and they are held together by C1C4 glycosidic linkage. But if you go to amylopectin, I can say this is an insoluble. Please understand, it is insoluble. Amylose was hot water soluble. It is insoluble in hot water and it constitutes the major part, 80 to 85% of starch. And we can say it is a branched chain polymer. The other one was unbranched. So it is also made up of alpha D glucose units and they are held together by C1C4 glycosidic linkage, but there is an extra branching, or the branching occurs due to C1C6 glycosidic linkage. Alpha C1C6 glycosidic linkage, and this C1C6 glycosidic linkage is absent in where? Amylose. Okay, so we'll see the structure, you will understand. So this is fundamentally amylose, this is alpha D glucose. This is alpha D glucose. So what is happening here? The C1 part of the carbon 1 of the 1 glucose unit is combining with the carbon 4. How? Because here initially the OH group was present. Here also initially the OH group was present. During the reaction, it will remove water like this H2O. Then what will be formed? An oxygen will come in between. And this type of linkage is called glycosidic linkage. Glycosidic linkage. Okay, so we can see it is a repeated alpha D glucose units combined together with in a straight chain with the glycosidic linkage. You can see that this is fundamentally our amylose. Okay, now when we go to amylopectin, again you can think like there is an amylose unit like this, okay, going like this, and in addition to that, carbon 1 of one amylose unit may combine with carbon 6 of a different chain. This is a different amylose chain. This is a different chain. So these two are getting branched like this. Again, like here OH was present. Here also OH was present. So water is removed and the oxygen is coming in between. Okay. So again, it is a C1 and C6 linkage. This is also an alpha linking. This is also an alpha linking. Okay, so C1 C4 link is also an alpha link, and C1 C6 link is also an alpha link, but here branching is occurring, like one chain is linking with the other like a branch. In terms of a branch, already linking happens via this unit. This is present in amylose also, but here in addition to that, one link, one chain, one linear chain joined with the other through C1 C6 linking. Okay, that is what is the branching, extra branching that is present in amylopectin. Clear? Now, if you go through the options, we can easily say the difference between amylose and amylopectin is, I can say amylopectin have 1,4 alpha linkage and 1,6 alpha linkage, which give rise to branching, Okay, so I hope the answer is clear for you. So my answer is option 1 would be my correct answer. Clear? That's it. Okay, so let's switch to the final question for the day. Okay, so the final question for the day is again from proteins. Okay, in a protein molecule, they are asking how the various amino acids are linked together. This is very important. So various amino acids can also link together through a different bond that is not glycosidic bond, that is called a peptide bond. I will explain you what is this peptide bond. Don't worry. Okay, it is an amide bond, I can say. So we can say proteins, they are fundamentally polymers of alpha amino acid. This is alpha. Okay, this is alpha amino acids. Okay, and they are connected to each other by peptide bond or peptide linkage. So what is peptide linkage? It is an amide linkage formed between a carboxylic acid group and an H2 group. So we will see one example here. Listen, so this is fundamentally a simplest amino acid, and that is called glycine. Okay, and this is alanine. Okay, so alanine is formed when R group is CH3. This is alanine when R group is CH3. When it will become glycine, when R is hydrogen, this will become glycine. So when these two are reacting, what is happening? The water is eliminated like this. Like I can say OH from the carboxylic acid is removed with hydrogen from the NH2 group. Okay, one hydrogen would be removed. So what you will get, H2O would be eliminated and there is a CONH. CONH2, we know it is amide. So one H is replaced by this, means it's a substituent of amide. Okay, so that's why the CONH is called peptide linkage. So this is called glycyl alanine. Okay, so this is how different amino acids are linked together. Clear? So I hope this is clear for you. Any doubt? Okay, so I can say the answer here in this case is peptide bond. I hope you have enjoyed today's important questions. So tomorrow also we will discuss some more important questions from the same chapter, and we will catch you tomorrow. Thank you.